I'm Gilda Barabino, president of BMES, and my job right now is to welcome you. Thank you for being here with us. I'd also like to up front acknowledge and thank Michelle Ciappa, who's done a tremendous job in organizing this session. And I'd like to thank my co-organizers, Drs. Yee and Sambanis. And I wanted to just tell you a little bit, real briefly, they're going to speak to you as well, about how this session evolved and why we're so happy that you're here and that BMES is able to work with the National Science Foundation to sponsor such a session. We recognize that there is a need to help develop our young people, those who are looking to get grants, and also a way to share and highlight some of the exciting research that's being funded by NSF. So what we decided to do was to come up with a session that we could both highlight the exciting work and help develop the new talent and share with the emerging folks some of the ideas around how do you best get grants, but see some of the ones who have been successful in getting them, and in a format where we could share with each other. So this format that we're using to first share some research with each other, break a minute, have some networking opportunities, and then after that have a more focused session on grant writing is something that we thought a lot about, and it's evolved. Last year was our first time trying it. This year we've done some improvements based on feedback that we received from our attendees. So this is a win-win. It's a win-win for BMES, it's a win-win for the National Science Foundation, and we hope it's a win-win for you as well. So again, welcome. We're looking forward to a very exciting session, and I'd like to have Dr. Yee join me next, and you'll hear a little bit more about where we're headed. And again, we're looking for any feedback from you for future sessions. I hope you like your thumb drives that we are providing for you. That was one of the, in response to some feedback we received last year, that would be helpful to have presentations, information in one spot. So that's one of the things that we would like to do to move forward. So, Dr. Yee, would you come and welcome as well?
Okay. Thank you. So I'm going to add my welcome to you. So as Gilda mentioned about, and I was 2 years ago I was the program director in the BME program. And, we talk about, and also Dr. Sambania with Sri, we talk about how we can help and as a junior faculty to apply the career award. And also I share with you my personal experience when I load my first career award, and nobody has guided me, and I asked for $1.4 million in the budget. And when I received a critique back, and I share, those comments with my senior faculty, and the senior faculty, the first word I heard from them said, "Are you crazy?" So, and that's basically, when I was become on the took on the program director. So I thought, we need a guidance, we need a help. The junior faculty is at least to share the experience. Of course, that's the purpose for these sessions. And you will hear on there's a panelist. They are very successful. They're career award awardees. They share you with their experience, and also you will hear on the tutorial information about a program from Dr. Sambania. So, and also I will offer you, and since I'm right now also the chair of the department, and I have the program director also has a. Although I have never received my career award, I'll be honest to you, but I have, it's the very experience. I serve on many panels, and also both on the NIH side and NSF side. And if you need somebody to help you to give you the feedback, you are welcome. So you have my contact information. You certainly, I will help you to read your proposal, give you the comments back, and again, so welcome you, and I'm so glad you're here. So then I hand the microphone to Dr. Sambania. That probably is the most and the person you want to hear from.
Thank you, Kai-Ming. So that makes two of us, because also I do not have a career award. Maybe it's a pattern among BME program directors. I don't know. But anyway, welcome to this BMES NSF session. So I'm Thanas Sambanis. I'm the BME Program Director at NSF. And Kai-Ming and Gilda did a great job initiating this session last year. So this year we think we're going to have an improved and expanded session. The purpose is twofold. One is to hear, as Gilda mentioned, the exciting research in science and engineering that is happening by the career awardees from NSF. So to hear from that, to see the type of research that NSF supports, and of course to become excited about the development in science and bioengineering, but also in the careers of the young investigators. The second purpose is that, although, as it was mentioned before, we're going to have a tutorial after this session, I still think that the best instruction is by example. So for the prospective career applicants, I think hearing the current career awardees present is going to be the best instruction that you can get. After this session, we're going to have a networking reception, which is going to be right behind this wall. Unfortunately, we do not have a door going through, so we'll have to go around. And after the reception, we're going to have the tutorial and the panel. So without further ado, let me start with the panel, with the presentations by the career awardees. The first one is by a new career awardee, Dr. Alan Dorval from the University of Utah, who is going to talk on manipulating neural plasticity to enable brain-computer interface learning. Alan?
Thank you. So let me make sure that this is on. I'm Alan Dorval. I often go by Chuck. My lab works in neuromodulation, and for this career award, we're moving into neuroprostheses. Here's my group. We're in the Department of Bioengineering at the University of Utah, and we've worked traditionally on deep brain stimulation, which is a therapy for a number of neurological disorders pioneered by Medtronic, in which an electrode is placed in a deep brain region, stimulated at high frequency to alleviate some neurological symptoms. This has approval in the U.S. for essential tremor, Parkinson's disease, dystonia, and obsessive-compulsive disorder. My lab works on understanding the mechanisms of DBS, why this therapy is effective, and potentially improving the therapy with newer devices or new programming paradigms. We focus on Parkinson's disease because it's well understood relative to some of those others. PD is characterized by slowness of movement and tremor, and it affects people in all walks of life. There are over 1 million Americans diagnosed with Parkinson's disease right now, and it's growing in our rapidly aging culture. We work in rat models for the most part. Here's a little rat. We implant electrodes into his brain so we can record neural activity and stimulate to alleviate some symptoms. And then we can inject a drug that provides some neurological disease. So in this case, we're injecting a drug that lesions this region here. You can see the cells on this side are stained black, and they are gone over here. So that animal has Parkinsonism on half of its body. And then we can train them to do some tasks, poke a hole for reward, or just put them in a cage and watch them over time. We can track their movement. So this is supposed to indicate the rat's here moving around, and we're tracking where he is. And this particular rat rotates to the right. This is his direction over time. He has Parkinson's disease on the left side of his body. So his right side is fine, and he tends to rotate like this. So in that 5-minute interval, he rotates 8 times. And that symptom can be treated with deep brain stimulation. We record from neural targets throughout the brain, but primarily in the basal ganglia. So these 6 structures here are sitting deep in the brain between cortex and thalamus, but not as a part of the thalamocortical motor loop. So this loop on the outside is what controls movement, and these guys in here sit and sort of look at what the brain is doing and look at what the cortex is doing and provide feedback. And we're not clear exactly what that feedback does, but if we look at the firing rate of these neurons, They certainly change as animals go from healthy to Parkinsonian and with deep brain stimulation. And these changes correlate well with therapeutic outcomes. So the firing activity of these neurons is associated with movement, but these structures aren't necessary for movement that we have learned before. If you know how to ride a bike or play the piano, and then you lose some of these structures, the majority of the output of these structures, you can still do those tasks. But what you can't do is acquire new tasks. So we hypothesize, and this isn't my hypothesis, this is widely held in the field, although difficult to prove in a concrete way, that these brain structures modulate the plasticity happening in the cortex. So that as the cortex is doing something, these brain structures report to the cortex if that is right or if that is wrong, and the cortex then changes its assemblies to perform these tasks better in the future. So given that, thinking about moving into central nervous system motor prostheses, for patients who've lost control of their body below the neck or below the waist, or from paralysis due to stroke or spinal cord injury, you can record fields of neural activity and use those fields to control simple aspects of robotic movement, but you're limited in the degrees of freedom that you're ever going to get from fields. If you record individual units of activity, neurons, hundreds of neurons, thousands of neurons in the brain, are all largely independent, and you can find some that fire quickly when an intention to flex an elbow or flex a hand or extend an elbow. And so people have done this in these two patients. This is an example from Brown University, and this is an example from University of Pittsburgh, where these women have not moved volitionally in a very long time, tens of years, but they're controlling a robot arm to give herself a drink of coffee here and a bite of chocolate in this case, via very complex computer decoding of individual neurons to robotic motor assemblies. But that coding has to happen every day or at least every week because the neurons you record from change with time. So while it can work really well, it requires a lot of programming for each event or each day. And it's something that's going to be difficult to implement in the long term. So what we imagine is instead of recording from single units, take single unit activity that we know corresponds to volitional activity and use that to program little assemblies to fire together. And if we can program assemblies to fire together, we can record the field activity of those assemblies. So this is a computer model example. We imagine the brain moving to doing something to flex an elbow. Some neurons fire in response to that. This little gray signal here is that neuron firing. Generally, we would detect that spike and move the robot arm. But instead, we detect that spike and co-stimulate the basal ganglia, which modulates plasticity, and the motor cortex, which is responsible for motor commands, to reprogram the motor cortex to work as assemblies so that a whole bunch of neurons fire together. And you take the average of all those neurons in a small group, and you get some field event that you should be able to record for years. So if you can record that field event for years, you can use that field event to control the robot arm, and it should be robust for a very long time. So this is, great in a computer model. In an anesthetized rat model, we've shown that we can do this to some extent. We have 8 electrodes in the substantia nigra reticulata. This is one of the output structures of basal ganglia, and in motor cortex. And we are stimulating most of these channels in substantia nigra reticulata with just a background pulsing, just kind of chronic, regular, boring activity. But we take one particular channel and burst it, drive it really fast, and then give it a pause. Drive it really fast and give it a pause. And we hypothesize that these bursts of activity are somehow signaling to the cortex that they should change their connectivity, strengthen their connectivity. Sorry. At the same time, we're stimulating cortex with different phase delays with respect to these bursts. So some regions of cortex are getting stimulated just before these bursts happen, and some regions are just after these bursts happen, to see if we can strengthen the connectivity around any one of these targets. Okay? So for different burst patterns into SNR, we can see the field event evoked in blue before this neuromodulation induction and after the neuromodulation induction in red. You see, for the burst patterns we use on this electrode and this electrode, we strengthen the field events everywhere in motor cortex. And in this electrode, we weakened it everywhere in motor cortex. So this is typical of what we see in our example experiments. We're still working out the burst patterns and the time delays that lead to effective strengthening or optimal strengthening. But if we can figure out how to optimally strengthen these groups, I shouldn't say optimal, how to strengthen these events into signals large enough to record, then we'd go into awake and behaving animals, where that field can be used to do something like, activate a food dispenser. So the rat can use this little field, which he can self-generate, to get a piece of food. So that's the idea of the career award that was funded. And it's only been funded for a couple months now, so it hasn't gotten very far. The educational goals of this award, I wanted to tie them in with the research goals, but it was difficult with closed-loop feedback, neuroplasticity, and mammalian systems. So we thought about moving into invertebrate models, say a cockroach, where you can stick a wire into the nerves that control his arms and his legs and either record or stimulate those really easily. I'm running a little low on time, so I won't get into too much of the details, but we have 3 environments in which we're going to do this. The first is in a computational neuroscience class for graduate students. We've got a couple different labs of this sort that they're going to try out. The labs that are most successful are going to be moved into an undergrad lab that I teach every fall in biosystems. It's about 60 students, all of our bioengineering undergrads. At the moment, there is no biology in our biosystems class. It is a systems and instrumentation class. So we would like to introduce some biology. Vertebrate biology is complicated, but I think invertebrate control is something that the class can handle. And then the most successful students at that will be shipped into a brain week awareness program that our program in neuroscience runs for junior high and high school students 1 week a year. And at the moment, it's largely lectures and manipulatives for junior high and high school students to play with, to learn about the cool things in the brain. But this will be learning about actual science in the brain, the things that scientists do. It's a doing thing, not just a sitting there learning thing. All right, so I'm out of time, but that's good. This is it. The career award's just getting started, so nothing has really progressed yet. But there are synergies in the lab looking at these things. One is closed-loop control. So this is something that needs to be done for this project to work. We need to be able to detect motor cortex activity and then co-stimulate in response. And we're doing that now, but in a different paradigm in closed-loop stimulation for deep brain, for Parkinson's disease in rat therapy. And then also stimulating multiple regions down in substantia nigra reticulata. This is a deep brain region. And if you want to stimulate tens or hundreds of different regions specifically, you need an electrode with tens of hundreds of different contacts. That's easy in cortex, where you have something like the Utah electrode array, but it's more difficult deep down, where you can't just shove 100 electrodes deep down. So this is a technology that we've put together that we're testing to let us do just that. So I thank the NSF for the Career Award. And some of this work is funded or has been previously funded by NIH, DARPA, the Utah Science, Technology, and Advanced Research Initiative, and Medtronic. These are the people of my lab, and in particular, Silvana Schister, who graduated last year with her master's, is the student who was pushing the work that has become the Career Award. Okay, thank you.
Thank you again, Alan. I think it's good that we use the 2-minute break for questions, and so we're going to move to the next presentation by Chris Jewell from the University of Maryland, also a new career awardee. His title is Dissecting the Role of Biomaterials in Lymph Nodes to Study the Shape Immunity.
Thanks. Happy to be here and have a chance to tell you about some of the work we've been doing. And I'm going to be talking to you about lymph nodes, which is what you see on this first slide. I'll try and tell you about the Career Award and what we're actually doing, and then also how it's impacted the other parts of my lab. So I've had the Career Award for about a year, and it's really focused on lymph nodes, which are actually, they're the tissues that control and orchestrate immune response. And so if you want to generate a response against an antigen or a virus or a vaccine, what happens is a piece of that foreign pathogen, a virus, bacteria, needs to make its way to lymph nodes. So in the lymph nodes, immune cells called dendritic cells, or antigen-presenting cells, they process and present that antigen to T cells and B cells that are in the lymph nodes. So these cells are actually already specific for the antigen, and when they see that antigen with the right signals, they expand and proliferate. And those are the cells that then migrate out and cause different kinds of immune responses. So we have T cells that can do things like destroy your own self cells that are infected with a virus to prevent the virus from replicating, or B cells that can secrete antibodies, and the antibodies can bind extracellular pathogens like bacteria to prevent those from circulating or infecting other tissues. So this is really a problem of kinetics and location. And so the lymph node and the cells in those tissues coordinate all of those responses so that what happens in these local tissues that swell when you get sick and all those kinds of things, and that controls what happens in terms of the systemic response. So what we've been looking at in my lab is really to understand how biomaterials interact with lymph nodes locally, and then how that causes different kinds of systemic immune responses. So this is obviously relevant for biomaterials because we've got polymers and complexes, we've got nanoparticles, we've got biomedical devices, and these things are being used in all kinds of different immunotherapies and vaccines because you can do things like load these up with different vaccine components or signaling molecules and then deliver them together or control the release of these vaccine components or protect your cargo. And so there's a lot of use of biomaterials in immunology. What is interesting is in the last 5 or even 10 years, there's a lot of work showing that materials that we thought were inactive, that were just carriers, like PLGA or polystyrene, they actually have intrinsic function that activates immune pathways without any other signals. So the carrier itself is actually playing a role in what kind of immune response you generate. And the career award is basically focused on understanding what properties contribute to that. So this is a resting lymph node. You see that the T cells and B cells, they're sort of segregated. Whereas in an active lymph node, you've got this intermingling of T cells and B cells because those cells, they have to work together to generate specific and high affinity or strong immune responses. So we've developed a platform where we can introduce biomaterials with specific properties or concentrations directly into lymph nodes. And we're using that as a platform to study how these materials with different properties impact the signaling in these tissues. Something that would be hard to do if you injected in your arm and couldn't control exactly what's making to those tissues. So there's some references down here if you want to read more about the details, but I'll just give one little snapshot. In the career proposal, so this is sort of one of the more fundamental projects in the lab, we're looking at 2 categories of polymers that are well-defined. They're easy to manipulate properties like the charges, the stabilities, the molecular weights, combinations with or without different immune signals. And I'll just, sort of the goal here is understand how the properties of the materials in the lymph node contribute to different systemic responses. So one of these that I'll just show a snapshot, stability. These are 2 different polymers, PLGA and something called polybeta amino esters that are widely used in vaccines. They degrade at contrasting rates. So PLGA, weeks and months. This other polymer, hours and days. Okay? So if we look at PBAEs, if we incubate that in buffer and it degrades over time, we have different fragment sizes, and we can introduce those into lymph nodes or to immune cells. And in this example, we take dendritic cells from mice, bone marrow, or from the spleen, and we incubate those with these different molecular weights of our polymer that is, that's what's happening as you deliver these, and they're degrading in a mouse or in a person. And we see a correlation between the molecular weight and the activation of the immune cells. Okay, so this is just in vitro. There's no other immune signals, just the polymer carrier. And in vivo, just one example of that, if we treat with the soluble polymer at a particular molecular weight, we get lymphadenopathy, which is swelling of the lymph nodes. So we get immune cells proliferating and infiltrating. If we put those in a different form, a particulate form, we also, again, without any other immune signals, we see that there's even a greater impact on lymph node activation. So this is sort of what the idea of the career is focused on, understanding how the materials people are using as biomaterial vaccine carriers, their properties, how they impact immune signaling, and then could we think about ways of using this information to rationally design new carriers that they're not just a carrier, but they actually actively direct the kinds of responses that we can generate. So, this is sort of the fundamental side, and then the career award's actually helping us learn new information that we're applying to other projects in the lab, and I'll show one example that's a little bit more translational. And in this project, what we're looking at is trying to control locally the lymph node environment to promote tolerance, immune tolerance, to treat autoimmune disease. So autoimmunity, this is like multiple sclerosis, type 1 diabetes, lupus, arthritis. And what happens is the body attacks a self molecule as foreign. So like in multiple sclerosis, myelin, which insulates our neurons, that's recognized as foreign. So T cells will migrate in, they'll attack that, and that causes loss of motor function. For patients, typically you'll get something that is an immunosuppressant. So it might block T cells from going into the brain, which, these things have been tremendously beneficial for patients, but it may also prevent T cells from going, some site of infection that you need an immune response. So your can be immunocompromised. So there's a really interesting idea in the field now that could you generate immunosuppression that's specific to myelin. And one way you could think about doing that is generate certain kinds of immune cells that are still specific to myelin, but when they see that, they respond in a different way. And then that way is to secrete anti-inflammatory molecules or to directly attack or inhibit the cells that are attacking myelin. So the idea is that you get an antigen-specific tolerance, but you leave the rest of the immune system intact. Right? That's kind of like the dream idea for autoimmune therapies. And of course, we're using biomaterials to do that, right? Because that's what our lab is studying. So these are particles that we've loaded with myelin peptide, and we're looking at a number of different tolerogenic signals that have been shown to promote tolerance. The idea is that could we control the release of these signals in lymph nodes and do something to help combat the side effects and the symptoms of autoimmunity? So we'll see if this... Okay, so this is a mouse that has multiple sclerosis. This is untreated, and this is kind of the gold standard in the field. What you do is you give a high dose of myelin with a strong inflammatory signal. That's to arm T cells against the myelin. And then you give a toxin. Let me replay that one more time. You give a toxin that opens the blood-brain barrier so those cells that are primed against myelin can go in and attack. You can see this mouse has lost complete function in its hind limbs, has limited tail function. Ultimately, it could become quadriplegic and would be euthanized. So this is a mouse that we didn't treat. We waited till it got as sick as they get according to our humane endpoints. Then we treated it with our lymph node depots, a single treatment at the peak of disease. This is the same mouse. So we've actually reversed the symptoms of the disease in this case. This mouse can actually stand up on its hind legs. She's not completely recovered. You can see the tail is still a little bit limp. So not full recovery of the disease, but we did pretty large groups, and using this aggressive treatment regimen, when they're sick as they get, we saw a really nice therapeutic effect. And so now there's a lot of work that's, again, tying back into the career work to understand what's happening in the lymph node that's allowing us to see these therapeutic effects. So that's one way I think the basic studies we've done has really helped us, in some of the applied stuff we're doing as well. So I see I have 2 minutes left, so I want to talk about one more thing that the career award has connected us to, and that's education and outreach. So when I submitted my career award, so I submitted it a little over a year ago. I got it in January, and we've been working on it since then. I put in that we were going to work with a high school that has a large fraction of underrepresented groups, and we were going to try and promote activities that would help give them exposure to STEM development in STEM fields. So it was like, we're going to do this, but it didn't have a specific research hypothesis. So what we said, we would create this program called the Program to Enhance Participation in Research, PEPR. And basically what we did is we have a group of students choose a topic at the beginning of the year. They're paired up with a graduate student or postdoctoral mentor, and they go through, for a whole academic year, a series of workshops and activities, like finding an expert that they're interested in a topic they're working on, contacting them, interviewing them, writing a research review article, designing and presenting a poster. And they get one-on-one feedback through the year. This is one example where they're working with their mentors at an in-person workshop. We also have a web portal we set up where they can get more frequent feedback from their mentors on their work products. And I think we just started the second year. We have this year about 25 students and 25 mentors. But from the first year, which ended in May, we had a really nice symposium where they all presented. They had their families there. We had representatives from Montgomery County Public Schools, which is the county the school's in. And what was also cool was the students built skills they can use. So they demonstrated, improvements in certain areas of STEM learning and technical communication. They also, got some line items for their resumes for college applications. They, had experience giving a public presentation, these kinds of things. And then one thing we didn't really anticipate is we got a lot of assessment data that we're now leveraging. So we are preparing a manuscript to submit on this that's education-focused. Last week, we submitted an NSF discovery research proposal. This is a 3-year proposal similar size to the Career Award, but completely focused on understanding how the activities we do impact self-efficacy, science literacy, and technical communication. So, a year and a half ago, I had no idea how to really think about any of these, and I probably know limited about that now, but we put a team together. We've got a proposal that we submitted, and this program that we're really impacting what is a pretty cool group of students. So for me, the Career Award has really been, even though it's only been a year, transformative in helping us do fundamental work, helping us connect that to translational work in the lab, and helping us connect to, doing outreach, but also using that to learn something about how the work we do is impacting those students' learning. So I just want to acknowledge, this is my lab. I've got a great group of people, funding sources, in particular, the National Science Foundation Career Award. So if you want to learn more, visit our website. And thanks so much to BMES and NSF for letting us present.
The next presentation is by Amina Kutub from Rice University, who is going to talk to us on virtual high-throughput model of brain microvasculature regeneration. Amina?
Thank you.
Thank you.
So broadly, my lab focuses on quantitative cell engineering. And when you think about it, there's two main ways of engineering cells. You can do it internally through things like synthetic biology approaches, or externally through matrices or through growth factors. And you can divide them into three types of stimuli: chemical, mechanical, and electrical. And our lab focuses on how do those interchange. How does a chemical, for instance, affect electrical response versus mechanical or morphological response? And our focus is largely on hypoxic factors, hypoxia-inducible factors specifically. And that's in part what the Career Award was about. So when you think of hypoxia, we take our response to hypoxia for granted. You're right now sitting here. All of your cells are normoxic, meaning you wouldn't be expressing compounds associated with what I'm showing you here. But if you went out and ran down the riverfront and you came back after 5 miles or so, one, you'd be tired, but your leg muscles also would be overexpressing hypoxia-inducible factors. And these factors, depending on what level of oxygen they sense, depending on what level of mechanical stress they sense, will expose the cells to a compound which allows, a dimer, which allows them to activate genes that are very quantitatively expressed as a function of the stimuli. And those genes could be involved in angiogenesis, inflammation, or glycolysis as examples. So if you did that training, if you went back and ran every day and you came back, over several weeks, you'd have new blood vessels in your legs. It's actually a wound repair process. But when you think of wound repair throughout the body and you specifically focus on the brain, it's a harder question. Your brain does have self-repair. So after a stroke, or even in Alzheimer's, you'll see changes in the hypoxia response pathways. But what you'll see is after a day, for instance, after an infarct, you'll see a small amount of growth, a bud. But as we age, and if the infarct's too big, your blood vessels, your small microvessels, can't repair enough to allow the area to have functional tissues, or functional nerves and functional vasculature. So our lab has been focusing on the quantitative aspects of understanding this process. And so when I first started, and when I was writing the Career Award, I looked at a whole bunch of images of blood vessels. And what you'll notice is that the microvessels, wherever you are in the body, are very different in their structure. So the skeletal muscle is very linear, the retina is honeycomb-like structure, and brain cancer is tortuous. So my question would be, well, okay, what's driving that? And if you look at these examples, one might first think, for the skeletal muscle example, it's mechanics primarily. And in part, you'd be correct. In this case, the vessel structure is aligning the muscle fibers. But I want to show you another example here. This is the same mechanical initial stimuli. These are images from Mary Dickinson's lab at Baylor, where on the left and the right, the only difference here is that you have different growth factors. And so you can have the chemical response driving the mechanics and the vessel changes. And that's what primarily we were interested in for the career. And we looked at, specifically, we wanted to understand how individual cells form these beautiful and elaborate structures, and how do they communicate with each other in response to chemical cues to form very different vessel structures. So we took an approach which combined computer modeling with experiments in the lab to answer those questions or start to address those questions. The computer model here, you can think of cells in this case as miniature adaptive robots. They're responding to their environment, they're responding to each other, they're branching, they're growing. And in this case, we're matching or we're ranking the computer models, every case, with what we're doing in the lab. And in the lab, what we're doing is we're stimulating an androgenic spheroid assay to have sprouting induced. And we're stimulating it by 2 growth factors present in the brain after a stroke, or actually even during exercise, BDNF and VEGF. And we're asking, with a cocktail of different levels of these growth factors, what is the outcome, and how does our computer model compare to that? And when I say computer model, it's actually tens and thousands of models. The unique way we approached this was by asking different hypotheses of what the cells could be doing. So we built a computer framework where you could say, I have a hypothesis as to how a cell would migrate, or branch, or divide, or proliferate. Plug that into the computer model and see how good your predictions, your hypothesis is compared to the actual experimental results. And that was what this framework allowed us to do. So in the end, what we got are predictions which were the best replicating what we could see in the lab. And these are two examples, the control assay and the stimulated case. And what you're seeing here, these colors correspond to cells migrating, branching, being idle, proliferative, or elongating. So cell behaviors. So this is very interesting for us. It was exciting because we could, on the computer, say, give these growth factors. You'd predict a certain stimuli, a particular response. You go back in the lab and you would be able to see that. That was exciting. But what it was qualitative in a sense, because the behaviors here are abstractions, and the environmental stimuli are what's driving the cell behaviors. We wanted to get a closer look. So what we wanted to do is get a molecular response from the cells. To that, we use the same paradigm of states. So the prior model I showed you had another innovation behind it. The cells were in what in computer science is called state machines. They remember where they were. So what they do in the next step determines where they were before. So they have a memory. When we started to look at this in more detail, we started to redefine how we define cell phenotypes. So no longer now are the states these broad behaviors. The states are dictated by what are actually seen in the lab. And what I mean by that is this. So we stimulated many, many thousands of millions of cells in the lab. We exposed them to the same combinations of growth factors we had in the 3D model. And we asked the question of what are the states are they in? Can you identify this cell as a unique cell, and how does it respond? So the color is not turning out very well here, but let me go forward and give you an analogy. We're asking the question. It's a complex question. We're asking the question of, for instance, recognizing a person as the same person, whether they're in different states or not. So I use the analogy to my nephew, who in these pictures is growing from year 0 to 3. He's also in different states. He's laughing, or he has yogurt on his face, or he's climbing the mountain. So you as a person who's very good, or humans are best at the best machines to recognize patterns, you can identify this as the same person, even though he's growing, even though he's in different states over time. That's a really complex computer problem to ask. So where do you even start? What you might start with is identifying Dylan, in this case, as in the computer to begin with. So what is the first step to that? The first step to that is literally recognizing him. But when you can see where we were starting off in the state of the art, we were quite a ways from where we were or needed to be. This is an example of dropping Dylan's image into Google Image Search, and you can see the variety of actors that are pulled up. So it's not really good. So we had to adapt existing technologies to get to a point where we could identify cells. So we have adaptive algorithms that, depending on the cell type you see, it will segment the cells, and it will give you a phenotype of cells. And so for us, we've been applying this broadly. So the Career Board really launched a program where we can apply it to work in the lab, but also clinically. So in these first two cases, we're working with both baseline models in the lab and also patient cells, Alzheimer's patient cells. And these last two, we're also working with human cells and patient cells. So for instance, let me show you what we do in each assay. Here you see neural progenitor cells as they're differentiating and as they're expressing different markers as they're becoming neurons. So we're capturing this information. We want to know how a single cell's phenotype is related into its network properties, and how those network properties are also related to electrical and functional properties. So for instance, for the vascular cells, we're asking a question of, can we identify regenerative phenotypes? Can we identify a combination of growth factors or molecular stimuli which would give us dominant phenotypes? And so we developed machine learning algorithms that you see the output of here, where we can say from a group of stimulated cells, we can pick out 4 dominant regenerative phenotypes. And we can program the cells to make a mix that we want to have as an outcome. And this is based off of profiling cells by things like fiber alignment, or the location in the nucleus versus the cytoplasm of HIF-1 alpha, a molecule of interest to us. We've also applied this to neural progenitors, where we're looking at the electrical activity as a function of the morphology and the local environment. So is a particular network structure associated with a particular electrical or chemical signal? In glioma cells, we're looking at glioma formation. And leukemic cells, we have a new program which can identify networks from biopsies of leukemic patients through a collaboration with MD Anderson. So every square that you see here is actually a network of proteins. And the colors represents which patients fall into which category. So for us, this is a barcode where we can use to give certain therapies for certain patients. It's been used right now for a clinical trial that's ongoing at Texas Children's for pediatric leukemia patients. So I just wanted to finish, I can see I'm running out of time, that the NSF career really launched a program. It allowed us to develop the tool set that we needed to start answering questions across domains. And it also allowed us to expand our educational section. So we have 2 outreach programs that have also gotten funded independent of the NSF career. One is for using all the images that you saw and making them into artwork. So these have been displayed at the Houston Health Museum. Right now they're an outstanding exhibit at Rice's Institute for Bioengineering and Biosciences. We also, this past year, have gotten involved in hosting a crowdsourced competition where that same leukemic data set that I showed you before is put out to the crowd of people. So all the different types of algorithms that can be applied to the data set can be used to predict outcome for patients based off of proteomic signatures and clinical signatures. We also developed new tools for them to interact with the raw data. And that's a new area where the lab's going. The images that you saw and the way you're interacting with the images actually inform algorithms. So we wanted to capture that in a systemic way. So instead of qualitatively getting your opinion of all of our analysis, right now every time you look at an image, we're capturing that and using it to inform our analytics. So with that, I'd like to thank, of course, the NSF and our other funding sources and people in the lab. I also didn't get a chance to talk about our collaborative work, but there's been a lot of collaborations which have been instrumental to the growth of the lab. So thank you.
Okay. So the next presentation is by Brenda Ogle from the University of Minnesota, who is also in the later years of her career award. And the title of her presentation today is Dissecting the Biological Impact of Stem Cell Fusion, a Unified Plan for Stem Cell Discovery via Accessible Engineering. Brenda?
Great. Thank you very much. It's fun to be here today, as was mentioned, to interact with the panel and also hopefully speak with you all later. So I was told to give a brief overview of my lab, what we're interested in, and then how the career integrated into or actually initiated many of these areas. And so we're interested in the study of factors that dictate stem cell state, and especially stem cells in the context of the cardiovascular system. And so this little schematic just shows all of the factors that many groups around the country and world are interested in. And I would say that the bulk of them have been focused on how growth factors, cytokines, chemokines, impact on stem cell state. And as an engineer, once we know those, how we can modulate them to control stem cell state. Also, gene expression has been used, another way of manipulating stem cell state to reprogram or program stem cells. We're, though, actually mostly interested in physical signals, so how the extracellular matrix impacts on stem cell state. And we've been developing matrices, three-dimensional matrices, that try to recapitulate the ECM with development. And the thought is, if we can do that, perhaps those signals are equally important and may augment or enhance differentiation protocols that utilize soluble signals. So what you're seeing here is a matrix, which is in red, and then these are cardiac progenitor cells that are seated inside. The blue is DAPI and green is the cytoplasm. Then our lab, as well as many others, after manipulating stem cells, have been looking at cell fate processes like replication, differentiation, migration, and apoptosis. But we'd actually like to add a cell fate process to this list, one that we've been studying and actually we think contributes or could contribute to programming and reprogramming, and that is cell fusion. So we've shown that stem cells can spontaneously fuse with mature cell types. We've been focused on fusion of stem cells with cardiomyocytes. And you're seeing here a particular fusion product. The nuclei are green because we use biomolecular fluorescence complementation to inducibly detect fusion products. And then what we've done recently, but I'm not going to talk about today, but will be coming out soon, is to look at the RNA transcriptome of individual single-cell fusion products to see what pathways are being up and down regulated to enable programming. So that's a lot of the other stuff that we do. What we did, or what I proposed in the career, was to actually look at new ways of monitoring stem cell state. You can see from this diagram that there are lots of things that can do that. And so if you're studying stem cells, you know that you have to monitor them constantly. And so what we started to look at initially was, can we identify new biomarkers indicative of stem cell state that don't require adding a label, probe, or a genetic reporter? And so that's one arm of the career. And the other arm was to try to develop technology that could actually detect such a biomarker in a meaningful way, perhaps detect it in a high throughput way, and also be able to sort cells based on that biomarker. So the big things are non-invasive and being able to sort or analyze in a quicker, rapid way. So here's where we started. I was working with a group at the University of Wisconsin-Loci who has many advanced microscopy systems, including custom-made multiphoton and confocal microscopy systems. And with multiphoton in particular, because of the high signal-to-noise, you can detect very weak signals, especially those corresponding to what we sometimes think of as autofluorescence or intrinsic fluorescence of certain molecules in the body. And one that piqued our interest was NADH. This metabolite is fluorescent. And it's also interesting in the context of stem cells because we know that stem cell metabolism changes with differentiation. We didn't actually know that until 5 years ago, and we conducted some studies that showed that this was in fact the case. This is Jane Squirrel. Worked in the lab, and she looked at differences in lactate production and also how stem cell differentiation changes if you force aerobic or anaerobic pathways. So in the interest of time, I'm going to skip over that, but just so you know, metabolism or the metabolic need does change in stem cells and their progeny. And so the question then was, does NADH change with differentiation? So here, Jimmy Fong took the lead, and he used a pluripotent cell line that was had a reporter for OCT4, which is a pluripotency marker. And so we looked at individual cells, and we tracked them as they lost OCT4 expression. And at that same single-cell level, we then monitored NADH intensity. And here are the sort of outcomes of that. On the top is GFP intensity, so this is OCT4, and we specifically picked cells that had high, medium, and low expression, and then we looked at NADH, which showed a significant decrease in NADH intensity with differentiation. This was pretty exciting to us, and we coupled it with not only intensity, but looking at lifetime of NADH, the time that it stays in the excited state. And we saw a similar decrease over time, and we also found that the component of the lifetime that was changing was that corresponding to bound NADH. So actually we're now following this up with studies that can link proteins that bind NADH and traditional metabolic pathways with emerging signaling pathways related to stem cell differentiation. But on the other track of things, we thought, now we've got a biomarker. Is there a way that we can look at it and use it to understand or at least monitor a stem cell culture? So we actually use this pretty routinely in our cultures, just statically. We look to see if the majority of cells are in the range of NADH intensity and lifetime that we have associated with pluripotency. But we also envisioned that we might be able to sort or purify such populations, and because time doesn't permit right now, we did further studies to look at whether or not NADH was predictive of differentiation of other mature cell types. And so also being able to enrich or purify based on those different levels of NADH intensity was of interest. And so the first thing you might think of, especially since I made these graphs to sort of look like a flow cytometry plot, you may think maybe flow would be a way to go. The problem with traditional flow cytometry systems is that you can only look at individual cells. Stem cells like to be in aggregates or large colonies. And so we didn't want to disrupt them to the single cell level. Also, traditional flow cytometry systems utilize single cell or single photon optics, which can't probe deeply into large aggregates. So what we did was simply well, it looks simple now, but at the time it was a good idea. We coupled multiphoton laser scanning microscopy systems with a flow system, essentially. And so we put a microfluidic device on the stage of a multiphoton microscope, and then in collaboration with LOCI, we developed software to be able to acquire and then analyze on the fly large particles and even single cells for NADH and other labels that we wanted to add to our cells. So this enabled us to interrogate the interior of large particles. We got enhanced analysis content because every single particle that passed by, we actually got a cross-sectional image of the entire aggregate. So we can view hundreds of cells at once. We could do large particles. They wouldn't get stuck. That was the on-chip fluidics. And then non-invasive acquisition was related to NADH and other autofluorescent molecules that we've been able to probe since then. We added a sorting component. Pedro Resto did that. We used it then to sort based on reporters, so extrinsic, we call it fluorescence, so bright things, and also on not so bright things, NADH intensity. And so this you're seeing, if we sort at early time points, embryonic bodies or stem cell aggregates based on this NADH intensity level that I was talking about earlier, at later time points we see an enrichment for EBs with a beating phenotype. And if we can also couple that with other optical parameters like scatter or size, and so we combine that with a size estimation, we can actually enrich even further. So that was sort of the culmination, and we're using this pretty routinely to look at stem cell aggregates, flow them through, analyze them quickly, and also purify. And we're hoping that because it's a simple microfluidic device, that eventually we can disseminate this to other labs that at least have access to confocal or multiphoton microscopy. I think I'm almost at a time, and I just summarized all of this. I did want to share with you some of the broader impacts quickly. This fluorescence image is not coming up, but in addition to stem cell biology, we think that the multiphoton flow cytometry system can be used for other things. So we've talked about using it to better analyze pancreatic islets prior to transplant. This image is just to show if you stain for dead cells under normoxic and hypoxic conditions, you see that in outer and upper and lower plane are not significantly different, but if you look in the midplane, that's where you see all of the cell death. And you might miss that if you were just looking at the cells on the exterior surface of an islet. The outreach kinds of activities that I proposed were built on a web that I tried to create with development of courses and new modules into a tissue engineering course and an extracellular matrix course that I was developing, and specifically to add the imaging component. And so by doing that, we were training graduate students to disseminate in a number of ways, and the most successful was through a summer science camp that YCell at that time was putting on. That's now evolved into different things. We also tried to establish a collaboratory that involved local industry. At the time that we started this, I'll be honest, it was too early. Most companies weren't that interested, especially in the Madison area where I was at the time. And so we had to take a step back and actually work through the Stem Cell and Regenerative Medicine Center at the University of Wisconsin, and that made it much more successful. So I'll just say that, the kinds of things that you propose are creative and new and things that you think might work, but as they evolve in your actual implementation of the proposal, they may change. And with that, here are my lab members, and of course, none of this would have happened if it wouldn't have been for the career award, which led to many, many other wonderful things. So thank you.